Hello everyone, this is Dr. A. Ramya, Assistant Professor, Department of Pharmacology, SPS Vistas. Let's see about cell and its organelles. Cell. Cells are the body's smallest functional unit. They are grouped together to form tissues, each of which has specialized function, example, blood, muscle, bone. Different tissues are grouped together to form organs, example, the heart, stomach, and brain. The human body develops from a single cell called the zygote, which results from the fusion of ovum, female egg cell, and the spermatozoan, male sex cell. Cell division follows, and as the fetus grows, the cells with different structural and functional specializations develop, all with the same genetic makeup as the zygote. A cell consists of a plasma membrane enclosing a number of organelles suspended in a watery fluid called cytosol. Organelles, literally small organs, have individual and highly specialized functions and are often enclosed in their own membrane within the cytosol. They include the nucleus, mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, and the cytoskeleton. The cell contents, excluding the nucleus, is the cytoplasm, that is, the cytosol and the other organelles. Mitochondria. Mitochondria are membranous, sausage-shaped structure in the cytoplasm, sometimes described as the powerhouse of the cell. They are central to aerobic respiration, the process by which chemical energy is made available in the cell. This is in the form of ATP, which release energy when the cell breaks it down. Synthesis of ATP is most efficient in the final stage of aerobic respiration, a process which requires oxygen. The most active cell types have the greatest number of mitochondria. Example, liver, muscle, and spermatozoa. Ribosomes. These are tiny granules composed of RNA and protein. They synthesize proteins from amino acid using RNA as a template. When present in free units or in small clusters in the cytoplasm, the ribosome makes protein for use within the cell. These include the enzyme required for metabolism. Metabolic pathway consists of a series of steps, each driven by a specific enzyme. Ribosomes are also found on the outer surface of the nuclear envelope and rough endoplasmic reticulum, where they manufacture protein for export from the cell. Endoplasmic reticulum. Endoplasmic reticulum is an extensive series of interconnecting membranous canals in the cytoplasm. There are two types, smooth and rough. Smooth endoplasmic reticulum synthesizes lipids and steroid hormone and is also associated with the detoxification of some drugs. Some of the lipids are used to replace and repair the plasma membrane and the membranes of organelles. Rough endoplasmic reticulum is studded with ribosomes. These are the site of synthesis of protein, some of which are exported from cells, that is, enzymes and hormones that leave the parent cell by exocytosis to be used by the cells elsewhere. Golgi apparatus. The Golgi apparatus consists of stacks of closely folded, flattened membranous sac. It is present in all cells but is larger in those that synthesize and export proteins. The proteins move from the endoplasmic reticulum to the Golgi apparatus, where they are packaged into membrane-bound vesicles. The vesicles are stored, and when needed, they move to the plasma membrane and fuse with it. The content are expelled from the cell. This process is called exocytosis. Lysosomes. Lysosomes are small membranous vesicles pinched off from the Golgi apparatus. They contain a variety of enzymes involved in breaking down fragments of organelles and large molecules, example RNA, DNA, carbohydrates, and protein, inside the cell into smaller particles that are either recycled or extruded from the cell as waste material. Lysosomes in white blood cells contain enzymes that digest foreign material such as microbes. Cytoskeleton. This consists of an extensive network of tiny protein fibers called microfilaments. These are the smallest fiber. They provide structural support, maintain the characteristic shape of the cell, and permit contraction. Example, actin in muscle cell. Microtubules. These are the large contractile protein fibers that are involved in movement of organelles within the cell, chromosomes during cell division, cell extensions. Centrosomes. This directs organization of microtubules within the cell. It consists of a pair of centrioles, small cluster of microtubules, and play an important role in cell division. Cell extensions. These projects from the plasma membrane in some type of cells, and their main components are microtubules which allow movement. Cell division. Cell division happens when a parent cell divides into two or more cells called daughter cell. Cell division usually occurs as a part of a larger cell cycle. All cells produce by splitting into two, where each parental cell give rise to two daughter cells. These new formed daughter cells could themselves divide and grow, giving rise to a new cell population that is formed by the division and growth of a single parental cell and its descendants. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. Types of cell division. There are two distinct types of cell division, out of which the first one is vegetative division, wherein each daughter cell duplicates the parent cell called mitosis. The second one is meiosis, which divides into four haploid daughter cell. Mitosis, the process cell used to make exact replicas of themselves. The mitosis is observed in almost all the body cells including eyes, skin, hair and muscle cell. Meiosis. In this type of cell division, sperm or egg cell are produced instead of identical daughter cell. As in mitosis, binary fission. Single-celled organisms like bacteria replicate themselves for reproduction. Phases of the cell cycle. There are two primary phases in the cell cycle. The first one is interphase. This phase was thought to represent the resting stage between subsequent cell divisions, but new research has shown that it is a very active phase, M phase or mitosis phase. This is where the actual cell division occurs. There are two key steps in this phase, namely cytokinesis and karyokinesis. The interphase further comprises three phases, G0 phase or resting phase. The cell neither divides nor prepares itself for the division. G1 phase, that is gap 1. The cell is metabolically active and grows continuously during this phase. S phase or synthesis phase. The DNA replication or synthesis occurs during this stage. G2 phase, gap 2. Protein synthesis happens in this phase. Quiescent stage, that is G0. The cell do not undergo further division, exits the G1 phase and enters an inactive stage. This stage is known as the quiescent stage, that is G0, of the cell cycle. Phases of cell cycle. The first one is prophase. Chromatin condenses to form visible chromosomes. The nuclear envelope begins to break down. Spindle fibers start to form and the centrioles move to opposite poles of the cell. The second is metaphase. Chromosomes line up along the metaphase plate, an imaginary line in the middle of the cell. Spindle fiber attached to the centromeres of each chromosome. The third is anaphase. Sister chromatids are pulled apart by the contracting spindle fibers. These now separate chromatids are considered individual chromosomes and move to opposite poles of the cell. The fourth one is telophase. The chromosomes arrive at the opposite poles. The nuclear envelope reforms around each set of chromosomes. Chromosomes begin to decondense into chromatin. The cytokinesis, the division of cell cytoplasm, typically begins during telophase. Transport across cell membrane, diffusion, osmosis, and active transport are some forms of transport seen across the cell membrane. Movement of substance across cell membrane. The content of a cell are completely surrounded by its cell membrane or plasma membrane. Thus, any communication between the cell and the extracellular medium is mediated by the cell membranes. These cell membranes serve two important functions. First one is, it must retain the dissolved materials of the cell so that they do not simply leak out into the environment. Second one is, it should also allow the necessary exchange of materials into and out of the cell. There are two major methods for moving molecules across a cell membrane, and it is related to whether or not cell energy is used. Passive mechanisms such as diffusion require no energy to function, whereas active transport does. In passive transport, an ion or molecule crosses the membrane and moves down its concentration or electrochemical gradient. The different type of transport mechanism across cell membrane are as follows. First one is simple diffusion, second one is facilitated diffusion, third one is osmosis. Diffusion. Diffusion is a spontaneous process in which a substance moves from a region of higher concentration to a region of lower concentration, eventually eliminating the concentration difference between the two regions. Simple diffusion. Transport across the plasma membrane occurs unaided in simple diffusion, that is, molecule of gases such as carbon dioxide and oxygen, as well as small molecule like ethanol, enter the cell by crossing the cell membrane without the assistance of any permease. Facilitated diffusion. This type of passive transport in which molecules that cross the cell membrane move quickly due to the presence of specific permeases in the membrane. Facilitated diffusion occurs only in the direction of the concentration gradient and does not require metabolic energy. It is distinguished by the following characteristic. The first one is the rate of molecule transport across the membrane is much faster than would be expected from simple diffusion. This is a specific process. Each facilitated diffusion protein transport only one type of molecule. There is a maximum rate of transport, which means that when the concentration gradient of molecule across the membrane is low, increasing the concentration gradient result in an increase in the rate of transport. Osmosis. Water molecule can transport through the cell membrane. The movement of water molecules through the cell membrane is caused by difference in the concentration of the solute on its two sides. Osmosis is the process by which water molecule pass through a membrane from a region of high water concentration to a region of lower water concentration. Active transport. Active transport is a kind of cellular transport in which substances like amino acid, glucose, and ions are transported across cell membrane to a region that already has a higher concentration of such substances. As a result, active transport employs chemical energy like ATP to move substance against their concentration gradient. This type of transport is commonly found in the small intestine wall and root hair cell. In this picture, we can see the different types of transport across cell membrane. The first one is diffusion. The second one is facilitated diffusion. These two comes under passive transport, and the fourth one is active transport, which requires ATP. Now we are going to see about cell signaling. It is a biological mechanism that occurs in cells which gives ability to revive or generate the signal in response to their surrounding environment. Communication between or within the cells. Cell signaling pathways is classified into two: intercellular signaling pathways and intracellular signaling pathway. Intercellular signaling pathway, it is a communication between different cells involving messenger traveling from a signaling cell to a target cell. The signaling molecules are secreted by one cell and bind to specific receptor on or in other cell, initiating a biological response. Intracellular signaling pathways. Intracellular receptor that are common types of cell signaling receptor located within the cell in the cytoplasm. Types of cell signaling. Cell signaling molecules are of the following type. The first one is intracrine ligand. These are produced by the target cell and bind to the receptor within the cell. Second one is autocrine ligand. They function internally and on other target cells, for example immune cells. Third one is juxtacrine ligand. These target the adjacent cells. The fourth one is paracrine ligand. These target the cells in the vicinity of the original cell, for example neurotransmitters. The fifth one is endocrine ligand. They produce hormones. Stages of cell signaling. Cell signaling takes place in the following three stages: binding of the signal molecule to the receptor, signal transduction where the chemical signal activate the enzymes, finally the response is observed.